Okay. So, good day, everyone. Thank you so much for joining this webinar organized by the International Ocular Circulation Society. We are very honored to invite Professor Tien Yin Wong to give us a lecture on ocular imaging for systemic diseases. I'm sure I don't need to actually introduce much, as Professor Wong is a world-renowned retina specialist and a scientist. So Professor Wong is now chair professor and senior vice-chancellor of Tsinghua Medicine, Tsinghua University in China, and senior advisor of Singapore National Eye Center in Singapore. So Professor Wong's research interests focus primarily on the epidemiology of eye diseases and the interrelationship between eye and the systemic diseases using advanced retinal imaging and the AI techniques. So without further ado, let's welcome Professor Wong to deliver his lecture entitled The Emerging Field of Oculomics, Historical Perspective, Current Status, and the Future. Yeah, let's welcome.
Okay, thanks very much, everyone, and I'm very pleased and honored to be able to give this talk online. Of course, it helps with travel and so forth, although it'd be good to have one day some face-to-face meeting to interact. But I'd like to thank Carol, Emma, Lisa, Yali, and others for organizing this online forum, and I hope that it's very successful and that many people can participate in collaborative efforts. Essentially, I see about 53 participants. I hope there will be more that joins. So I think maybe I'll just check, Emma, you are able to see the slides and can hear me. Am I correct?
Yes, we can see it very clearly.
Good. All right. So let me get started. And I think today I'm going to give you a talk on this topic, which is quite really exciting. And I have a lot of thoughts about this, but I'm going to focus a bit on really some of the historical perspective, because the historical perspective will give us the background of what we need to do in the future. Because this field, although oculomics is probably about 3-4 years old in terms of the terminology that's been used, it's really quite a lot of work has been done in the past, and there are some important lessons for us to look at. So I'll focus a bit on the historical perspective. Of course, I'll update a little bit on what we know now, some of the latest developments. Many of the people in the audience here has also participated in that research, and a little bit of thoughts about the future. So I want to start with me in Wisconsin, because in fact, I was introduced to this field in the late 1990s, and that's with Ronald and Barbara Klein. Many of you will be aware that they are giants in ophthalmology, particularly for the studying of retinal diseases, as well as using at that time what was the most advanced techniques, which is retinal photography to document and to quantify diabetic retinopathy and age-related macular degeneration. And that became known as many of you will be aware, the Wisconsin classification and so forth. And I think using very rigorous scientific methods and epidemiological studies and using data, it's very important, for oculomics as well as for any other fields. But what I wanted to show you was that during my time when I was doing a postdoctoral fellowship with them there at Wisconsin, they asked me whether I was interested in doing, research direction in using what is not common at that time, which is to use the eye as a way of studying systemic disease. In other words, using systemic disease as the outcome of the research. Now, this is quite different from many ophthalmologists who would use the eye diseases or the eye as the outcome of our method of studying. So you need to almost reverse the direction of research, right? And that I felt was a quite useful and important topic, very interesting, in fact, and when I studied it, I found that there was very little on that. Subsequently, I also was very fortunate to work with another giant in ophthalmology, this Paul Mitchell, who is famous for the Blue Mountains Eye Study in University of Sydney. And he also caught on to this very important emerging field, which is to use the eye as a way of studying systemic disease. Now, I think with their mentorship, Ron Klein and Barbara Klein, as well as Paul Mitchell, and many, many others, which I do not have time to mention, we started some of this work in the early 2000s. So what we do, right, this emerging field of oculomics, right, the eye as a window to systemic health and disease, as I said, we started around late 90s, and in 2000s, we were already doing some work and proposing some of these concepts at that time. And this kind of rapid review that I did with Lancet Neurology, I think, if I'm not mistaken, was the first few issues of the Lancet Neurology at that time, suggested that maybe it's something that the ophthalmic community can contribute, right? So not just to solve eye diseases, but to solve maybe a larger health and systemic disease. And, you know, in many of my talks, I've looked through a lot of my talks were in that area, essentially. So I'm going to cover three aspects in the next 45 minutes. First, why is this concept appealing, right? The historical perspective. Second, what are we doing? And I would want to move what I would say from qualitative to quantitative to what we are now using in the AI deep learning space. And then I'll look at what I think are the future and where do I see oculomics going, and how can we move the field forward. I think there are many, many research groups working on this now, and I do encourage them to work on it, but you know, that was not the case many years ago. So let me go back to the historical perspective, right? So when we talk about oculomics, it's really, as I said, you know, there's some fundamental observations that sometimes we take for granted, which is that, you know, when we look into the eye, or you look at the outside of the eye, but maybe really in the inside of the eye, the retina, the blood vessels, as well as the nerve layers, as well as what you can see here are individual cells with adaptive optics and with even more advanced wide field imaging. You're seeing actually all the way from structure to cells, essentially, right? They can be viewed, and more importantly, they can be viewed as well as imaged and measured and monitored multiple times over the course of a person's life without the need for invasive kind of methods. And I think that's something that we should not take for granted. Now, if we look at, as a retinal specialist, we already know some of these issues. And when we started this field, we know that there were these, what we call classic changes that are seen in aging, in people with atherosclerosis, but more importantly, in people with hypertension, particularly chronic hypertension. And these are what we'll call, you know, hypertensive microvascular signs, or hypertensive retinopathy for that matter. And when we look at these signs, they are not of interest to ophthalmologists, but they are of interest to this rest of the community. And even in the late 1800s, as well as in the mid 1950s and so forth, there were also observations that people with these obverse retinal microvascular signs were associated with poorer systemic health and were at risk of dying, right? And this became known as, for example, the Keith-Wagner-Barker classification of hypertensive retinopathy, something that we are at the moment, you know, take for granted, but it was really from these observations that started. Now, not just in the retinal microvascular signs, we also know that there were significant studies in the late 80s and in the 90s that when they were able to see the optic nerve, for example, measuring it from, you know, even fundus photograph, but then subsequently with OCT, that there were some observations that the eye was really intricately connected to different types of disease. And these are papers that sometimes we missed out because we are now moving into much larger data sets and clinical studies, essentially, right? But a very important concept that I would say started was the fact that different types of signals in the eye may point to different disease pathogenesis. And I just want to mention two, I think, very interesting observation, right, which is really from histopathological studies. So Christine Churchill is a very established, well-established ophthalmologist and also pathologist who has looked at, really histopathology with post-mortem specimens and so forth, and kind of correlating changes in the retinal ganglion cell layers with Alzheimer's disease, for example. And there were also other people that looked at retinal vasculature changes and correlated it with people with cerebral vascular disease. So we kind of feel that maybe different signals, if we were able to fine-tune those signals, would give us different ideas about pathogenesis. So that's also a very important part. Now, how do we look at oculomics as a framework, right? If we want to look at the natural history of disease, we can say that there are the traditional risk factors, of which age is one, but of course there are cardiometabolic disease risk factors, whether they are medical or environmental, or they are what we'll say more intrinsic, like genetic factors, as well as other newer factors. They do go through a process, and that process could be short, but most often it is long. In fact, there's been a whole idea that many of the chronic diseases start at birth and even prenatal, right? The whole idea of Barker's hypothesis is that a lot of our chronic disease is not just determined by the genes, but the epigenetic mechanism that starts getting environmental influences right from when we are conceptualized in the womb, right? And in early life itself. And therefore, preclinical disease could stretch what I would say over decades. And if we are able to measure preclinical disease earlier, then maybe we can prevent or treat clinical disease, which is just listed here later, right? And the methods that we're doing is what we'll say developing a whole range of tools, for example, carotid ultrasound, which measures large blood vessels in the neck, in the carotid, as well as in the brain. There's, of course, many different types of MRI, PET scan, and so forth, and in the heart, ranging from ultrasound to CT and MRI and so forth. Now, is it possible, therefore, that the eye signals is also able to tell us something about the long pathway of preclinical disease and act as, therefore, what we'll say is a biomarker or a predictive or a screening tool that allows us to predict the future development of clinical disease, right? So that's the kind of concept that we would like to use, essentially. So I think when we started this work with the Ron Klein's, one of our early papers suggested that you could really predict clinical disease. And in this paper, we found that various microvascular signs were predictive of incident clinical stroke, 3 years later. And that was, showing that the severity of the damage was correlated with the risk of stroke. Subsequently, there were also other groups looking at this, and this came out as a press release in 2005. Many of you would not remember this, but I remember reading it, and this was surprising because people assume that the eye signs always occurs after the disease development. So when there is what we call an eye sign, diabetic retinopathy, occurring in people without the disease, without diabetes, in pre-diabetes stage, it was a surprising observation, essentially. And when we looked at this, we found that it was not really that surprising, right? Obviously. And so one of the papers we've done was to say that even these eye changes that were typical of disease occurred at levels of disease measures that were not before the clinical disease development. So in this case, we can see so-called retinopathy signs in people without diabetes and in ranges really in a very low glucose level range, for example, less than 5, right? And that would obviously not be diabetes. So I think this suggests that the eye signs occurs maybe with subclinical disease development. Again, a very important foundational concept for the field of oculomics. Now, subsequently, there are many groups working on this rationale, particularly with regards to cerebral vascular disease. Carol Cheung, who's here, started working on this field along with people from Edinburgh as well as from UK and many other groups. And in Australia, we've looked at using this eye signs to kind of classify people with stroke and different types of substrokes, essentially. Now, I'm going to go into what I'll say the progress that's made. Now, we know that this concept is useful. We are now trying to say whether we can apply the first use of, I would say, computer science involved in ophthalmology in one sense, right? And in those early days, we worked with computer scientists who was before the machine learning and before, of course, the deep learning AI phase, right? This was very, I would say, rudimentary computer methods, right? Basically looking at pixels and seeing whether we can measure the mid-range of the pixel density, right, to measure vessels. But we said, let's quantify some of these changes rather than say, are they present or not, which is how retinopathy signs were classified at that time. And we then worked with some of the very early pioneers in this field, and that's Larry Hubbard, who looked at calculating some of these changes, which is what people accept now as the central retinal artery equivalent and the central retinal vein equivalent, but there were a lot of thinking behind it and a lot of experiments. And the original software, as you can see, was very crude. We developed much better software over time, and this is really a very nice color-based software that was being used in the MESA study, for example, which was one of the first study in the US that used digital retinal photography rather than film-based photography. And therefore, from there, we are able to now quantify what we say as summary measures rather than scattered measures. Now, I want to mention this, which is important because in the field of deep learning, we have very little summary measures. We have a lot of what I would say black box measures. In other words, we have no idea what they're measuring. And I think some of the field of oculomics should go back towards measuring things that we feel are explainable, right? The whole idea of explainable AI is for people, there are scientists and physicians and biologists to say, yeah, I can understand why. Now, I'll tell you why this is appealing, right? Because this summary measure here suggests that CRAE and CRVE, and you can see with your own eye, one is narrower than another. Now, it is not exactly identical, but nevertheless, the summary measures needs to be reproducible and needs to fit with what we're seeing. We are not seeing it in the oculomics field today. And I just want to mention this here for many of you who are looking at this, right? In other words, what are you measuring? This explainable phenomenon is even more important for oculomics, essentially. And we found many association studies with this, which I will not go into, but these were associated with many different diseases. For example, this is something quite surprising, which we have never discovered before, right? Which is that the narrower retinal arterioles were associated with the development or the predictive ability of diabetes over time. And is it related to, a vaso suppressive phenomenon? Is it related to hypertensive, chronic hypertension as part of the phenomenon? We are not sure. But what we know is that there was association between quantitatively measured retinal arterial caliber, and subsequently there were also some differences with venular caliber, which I will not go into, but they predicted the risk of diabetes over time. Of course, there was also this whole idea that they could predict cardiovascular disease, which has many, many papers, which I will not go into. But basically, what it does show is that retinal vascular caliber measurement and the separation between arterioles and venules, which provided different information, are provided what I'll say more precise than a yes, mild, no severity level of retinopathy signs, more quantitative assessment of CVD and stroke risk. And I think that the oculomics field must be able to provide that kind of quantitative assessment. The deep learning field, unfortunately, is still looking at many yes-no phenomenon because, it's, the way in which it is graded has not been made clinically useful and explainable for particularly clinicians. Now, I also want to mention is that it's not just vessel caliber, because when we started looking at blood vessels, we started saying that maybe there are other information that was useful. And the other information, of course, happens to look at branching and patterns and other phenomena. And this led us to work with computer scientists to look at things like fractal dimension, like tortuosity, like bifurcation. And, there are many such software in about the late 2000s, early 2010s, not just the Singapore software, which we call SIBA, but there was also Vampire and Quartz, which are used, for example, by the London group, the UK group, and they are applicable to many people who would like to use this. But I would still feel that these software, although they were useful, they have some limitation, which I'll mention. So this is a SIBA software that was developed, and we have kind of like used it for different types of association studies that you are able to see here. Now, I want to move a little bit about not just retinal photograph, because in the field of oculomics, this is other areas that has not been as established, and partly because there are much less data. Fundus photographs are more widely available and maybe more useful for ultimately for screening and for clinical prediction. However, I would say that OCT would be not far behind, because the day of cheaper, widely available OCTs will be present in many optometry and clinical practices. And of course, there would also be OCT and geography. But these are also very useful studies from the UK Biobank, Paul Foster's group, as well as Kamran Ikram's group, Caroline Klaver's group in Rotterdam, suggesting that OCT are also associated with different risks of dementia developing. So I think the confluence of factors suggests that the early observations of optic nerve retinal ganglion cell are being useful for what we'll say a neurodegenerative disease, and the retinal vasculature as more important for a cerebral vascular disease, including stroke and vascular dementia. I think that theory still has a lot of consistent studies showing, essentially. Now, what has also happened, of course, is this whole idea that what happens later, right? What does it mean? And when we did some meta-analysis, our own conclusion, which was suggesting that this view would hit a little bit of a roadblock, because the incremental predictive ability, in other words, is it useful as a tool, particularly for what I'll say for screening and for prediction, may be modest, right, and unlikely to translate. This was a conclusion that we had from some of the meta-analysis and some of these things here. And therefore, we were, I would say, this field entered a little bit of technological, you can say, valley, right, waiting for something to happen. And as I said, there was some technological breakthrough. And of course, the new breakthrough came through with deep learning, which I think everyone is quite aware of, which started about in the mid-2015s. And of course, the Google's paper and so forth started this whole idea that maybe you can go back into the field, which is now what is called oculomics, with deep learning and artificial intelligence. I'm sure you're aware the deep learning founders were all Nobel Prize winners this year, essentially, right? So if we go back to that, understanding that part here, are we able to therefore use this imaging techniques, particularly on the retina, but not just limited to the retina, and use it to predict what I'll say are three important frameworks. The first is to see whether it can help us look at risk factors itself, right? So that's one. So in other words, can we get a risk score of someone using the eye? And that risk score should be better than the current risk factors that we have, including, a person's age, a person's blood pressure, and so forth. The other is whether it can replace some of the more expensive and not widely available tools, right? So for example, if you want a CT of your heart and you want an MRI, I mean, you can't do it anywhere except in the major hospitals. And can the retinal imaging, whether it's OCT or fundus photograph, replace that? And the third is, of course, whether it can just detect the disease itself rather than replacing some of the tools and the biomarkers. So these are always what I would say the three approaches of oculomics, essentially. Now let's look at the first approach. Now, many in this audience would be aware about the Google paper, which was a phenomenal paper. When it came up, we were not certain. In fact, I reviewed the paper and I was saying this is really, quite amazing, right? And maybe it's the first time that was done. I'm not sure that many journal editors would do this, but the PEP wrote to me and said, what do you think of this? I said, it's phenomenal, but I would like to be able to test their algorithms, essentially, right? And they did send, we did where we externally tested with a reviewer, which is our study, the Singapore Epidemiological Eye Disease Study, right? To test whether their predictive ability was equivalent to another data set that has not been in their same lab. And this is really one of the most rigorous peer review I've done, right? Because we requested for that, essentially. And, the paper is quite well known, so I won't go into the details, but it kind of predicted quite well and was replicated, which we wrote an editorial on in the Singapore Epidemiological Eye Disease Study. So that proved, the concept that it really can provide a lot of signals. Subsequently, we also looked at this and we were now a little bit more disappointed because, rather than saying that it can predict everything, we have found that maybe it is not able to predict a lot of things, essentially, right? So, we examined a lot of markers and many of it could not be done. Now, why is the case? Can the eye provide information about everything? Is oculomics able to provide as much as genomics or proteomics? I do not know the answer. All I know is that in this study, we were limited by sample size. So if you have a larger data set, maybe you have more information. I think the extent of information on the eye likely is not the same as a blood, right? But, it will likely be more than what we are thinking it could predict. But there are now several studies that have kind of looked at association, and there are still a whole I would say, range of what I call traditional or non-traditional risk factors that could not be predicted by the eye, and that would not be surprising. So understanding, number one, in oculomics, what is the most consistent risk factors that could be predicted from an eye is a topic that I hope some of the students or other research here will look at because it's not clear, right? The Google paper suggests that it could do a lot of things. We think that it's probably limited, and what it can predict and what it cannot predict, I think, is quite important. I think many groups would have not reported on what they cannot predict, but we need collaborative studies that are large enough that will be for us to be able to ascertain robustly what the eye does not tell us, essentially. That's a very important part of the field. Now, in one sense, we went to age as another very important risk factors. And of course, there's a lot of search of what we call biological age. And there are a few groups working on this. Lisa Zhu, who's on this call, of course, works on this as well. And when we looked at this one is, of course, to just look at whether or not the estimated retinal age is useful, which means that the retinal age is beyond our standard chronological time-dependent age, our date of birth minus the, our current age, and that gives us our so-called chronological age. And then I think, of course, it's more, I would say, precise because, it has information that is not possible with just the time of your chronological age. The retinal age would have really congregated factors such as the level of blood pressure, your amount of cholesterol, and other so-called cardiometabolic diseases that we will have accumulated over time. Now, this very important and I think very interesting concept that Lisa and Mingguang He did was looking at this age gap. And I think the idea is also conceptually very appealing, which is that you're not just interested in the age, but the difference between what is your so-called phenotypic type of age, as well as what is your chronological kind of age. And this gap, if it is positive, is obviously not so good. But if it's negative, means that your retinal age suggests that you are younger, maybe you did a lot of exercise or you had a healthy diet and you're genetically protected, then you, in theory, will live longer based on your retinal age compared to a negative age gap compared to your chronological age. And I think that they've shown this quite elegantly in some of the studies, which I won't go into because there are many studies that this group has done. The second thing is, of course, whether it can replace what I would say are expensive, inaccessible biomarkers. Now, there's a lot of nice work that remains to be done, and that's another area of research that is, I think, has a lot of areas of innovation. One is that many of these biomarkers that have been studied in large studies do not have retinal images, right? So there are very few studies that you can correlate, for example, CT scan of the brain with a retinal photo, because there aren't that many of these studies, essentially, right? And in those they have done, this is a very nice study that looked at carotid IMT and a retinal image and, predicting a score. What they will say is a fundoscopic, or basically it's a retinal carotid score is what I would look at. And of course, it seems that this retinal carotid score is able to predict cardiovascular mortality just like carotid IMT would be, except that you may not need a carotid IMT, right, or ultrasound. It can be done purely in an optometry clinic without you needing to send it to a cardiologist or a neurologist. We've also done this with Rim Tyler and Cheung and others in this, whereby the data set that we had allowed us to predict what we'll call a coronary artery calcium score, which is quite commonly used in cardiovascular disease circles to predict the risk of cardiovascular disease. So getting a calcium score is something that some of us would know. For example, my calcium score at the moment is 0, so I think I'm still all right. But many people could be in 1 or up to 100, and so on and so forth, essentially, right? And therefore, having this retinal predicted or estimated calcium score, could it predict risk of disease, was what this paper was looking at. We have not seen many replication in this study because there's very few calcium score type of studies available. I still do feel that the amount of collaborative cross-validation study is another area that we have not been able to do so well. And therefore, individual models that came out is very hard to say whether they're accurate or predictive, or whether it be useful or generalizable to other populations. We went back to our retinal vessel measurement, and with Carol and with some of our collaborators, Winnie and Moonli Lee from the School of Computing in NUS, we set out to look at whether we could do a fully automated measurement of CRA and CRVE, and this is what we call SIVA Deep Learning System, SIVA DLS, and this allowed us to use it much more quickly and faster to measure these calibers, which were in the past semi-automated. In other words, the computer still required a human input for us to measure things. And this was used in studies such as this, which Carol and Chris Chen showed that the CRE and CRV measured this time with the deep learning SIVA DLS software was predictive of, for example, risk of cognitive decline and dementia, as you can see here. Although, if you look at the what I would say the predictive ability, it still remains to be seen whether it is clinically completely that useful, essentially. Now, I want to move on to another area in which they've started broadening the association. So instead of just looking at whether or not it's going to be associated with, what I'll say are biomarkers that are standard, for example, calcium score or carotid IMT, there is now something that whether it can look at genome, right? and there's studies that look at the association between retinal image using deep learning and genomic, you can say phenotype, or you can say genomic, the genome of a person, essentially. And there's also some association here, which I think suggests that this is an area that would be of interest. So the idea of oculomics linking with genomics, linking with proteomics, with metabolomics, and other omics in the body, in other words, other information systems, I think is an area of interest. Whether it is of clinical use, I think remains to be seen, as I will mention in the subsequently. So the last part, of course, is where we are now thinking about specific systemic disease. And specific systemic disease, there are many studies out there. I have no time to go through that. One very interesting paper, which went into Nature Biomedical Engineering, but I think some of these kind of association studies will be less and less useful over time, was on anemia, right? So I think what was done was that, can you predict the presence of anemia, which typically requires a blood test, using a retinal photo? And you can see here the accuracy is good. It's about 88% or more, but whether it is useful, practical, and who will use it remains to be seen. So anemia is one of the studies, and then also there's kidney disease. There's several papers on this with this group with Chinyu and Chairu, who is working on this field extensively. And then there are also others that have looked at type 2 diabetes as well as diabetes complications. So many specific disease without needing to go into what I'll say risk factors or intermediate biomarkers, but going straight to clinical disease has already been done. So again, there are many papers out of this there. One very useful, and I think I would say attracted quite a lot of attention, because it is very difficult to diagnose Alzheimer's disease without, I would say, expensive and even invasive techniques, right? Expensive means you need PET scan, you need MRI, and invasive means that you might need lumbar spinal fluid, right? Cerebral spinal fluid, CSF, to diagnose Alzheimer's disease, particularly in the era whereby we have very expensive drugs, and these drugs are only useful in early stages of cognitive decline in Alzheimer's disease. So how are you going to screen them? They cannot be all brought to the hospitals, right? For you to screen these patients, right? They are likely to be in a community, and which neurologists or GPs will be able to diagnose what is you can say early cognitive decline or mild cognitive impairment, MCI, or frank Alzheimer's disease, I think is still lacking. So in the community assessment of screening and of managing Alzheimer's disease, there's a major gap there. And I would also encourage that the researchers and the students working in this area concentrate on screening for things that would require early treatment, and yet the current way of screening requires expensive tests like a CT or MRI, right? So there's clearly a gap here that Oculomics could fill, right? It may not fill many gaps, but this is one gap which I think fills. Expensive treatment, but the treatment works in early disease, and yet the screening of early disease, which occurs in the community, cannot be done with existing methods, right? That's quite different, for example, in cardiovascular disease. Cardiovascular disease, in some sense, can be predicted by simple tests in the community setting. Alzheimer's disease cannot. It's also the case for diabetes. Is it useful to predict diabetes early? Yes, it is. But why do you need it in the community when it can be done by you can say a relatively simple urine or blood test, right? But Alzheimer's disease, you need it in hospital setting. You need neurologists. And yet the treatment for it is so expensive that it's useless to diagnose late Alzheimer's disease and give, a very expensive drug. So I think understanding the clinical needs is very important for the field of oculomics, where there's where that it could address a specific problem. Now, I'm going to end this talk by talking a little bit about the future. Now, what are we going to go into? All right. As we all know, and I've kind of shown this slide in many different scenarios, right? Bringing multidisciplinary teams together are not easy because, the scientists and the clinicians sometimes don't understand each other, right? And obviously, right, the training is different, the setting is different. A lot of our engineering scientists, a lot of computer scientists work in engineering labs in universities. Our clinicians work in the hospitals, understand the clinical gap, but bringing the clinical problem and the technical solution together requires that kind of interdisciplinary teams, which is common, but much more common now, but it's not that easy, essentially. And one of the things that I do want to mention that some of you are aware, and many papers are out there, many of these major papers in very high impact factor journals, for example, are highly cited, but often do not make it to the translational pathway, partly because it needs a very clear understanding of where that technology fits and where it will be used by people, right? And this is not that easy. And I think that translational gap is something that I do want to encourage, again, all our students and postdocs and other researchers here working on. I think this forum allows you to bring some of these teams together, essentially. There's also a roadmap for looking at biomarkers. So if oculomics is going to be a biomarker for certain systemic disease, then it needs to go through that pathway. Now, what's that kind of pathway? Now, in cardiovascular and stroke, there are, and Alzheimer's disease, there is a pathway, right? And that pathway means that it needs to go into what we call a framework, preclinical exploratory studies, basically, experiments, right? Algorithms development, right? And then understanding using retrospective studies, and we have many studies in this area, right? Retrospective, and then prospective diagnostic accuracy studies and disease burden reduction studies. Does it reduce the burden of disease, the incidence of disease, the morbidity of disease? I think they're much, much less, essentially. And so if you look at some of the examples of this, this is what a lot of societies and governments would like to see, right? So we have proof of concept, we might have proof of principle, we might have proof of efficacy, we might have proof of retrospective studies that looks at accuracy, but do we have a proof of prospective effectiveness? Is it reproducible? Is it generalizable? And lastly, is it feasible? Is it implementable? I think those kind of things Oculomics needs to go into that thing. So I think I'll go into this whole idea now. Piers Keen and Siegfried have proposed the term of Oculomics, which I think is very important. I think what you would see is that most studies at the moment are association studies, single disease studies, studies that have not had many rounds of replication, partly because a lot of the algorithms are not either open source or are not easily applied, and there are many issues there. Then on top of that, I find that there are still a lack of real-world prospective research. In other words, for example, let's say Carol Cheung's Alzheimer's disease detection, has it been tested in the clinic in a prospective real-world setting? So you screen 1,000 patients, 10,000 patients, how many have Alzheimer's disease, how many have you missed, how many have you diagnosed? Do we have those kind of studies? A very important part, which is not the same for other parts of AI, for ophthalmology, is that the user of this technology typically is not ophthalmology, right? And even though I put ophthalmologists and optometrists here, why would ophthalmologists be interested in whether or not a patient has cardiovascular, diabetes, or Alzheimer's disease? We are not experts in this. We certainly do not know the guidelines for treatment or screening or management. So who's going to use this is quite important. And can it be used by general practice? Can it be used by groups that are interested in screening? We have not identified that group more. Now, what about the algorithms itself? Does it meet the screening criteria? An AUC of 85, it's nice to use in a publication, but what's the risk involved if you have false negative? What's the burden to the clinic or to the healthcare system if it is false positive. Now, we have done AI for diabetic retinopathy screening, and I would say that many of our clinicians were unhappy about having too much false positive. In other words, we were referring a lot of what was not diabetic retinopathy to add to the burden of ophthalmologists seeing patients that the AI said, you have diabetic retinopathy, go to your ophthalmologist, and they say, actually, you don't have diabetic retinopathy. So in other words, this creates a very negative cycle of understanding of using technology. So a very good balance of not just AUC, but having low false positive and low false negative rates are important. Many Ministry of Health and other officials would like a very extremely low false negative rates. In other words, they do not want to miss out on disease, but not knowing that when you tweak it and you have a lot of false positive that goes to the to get referred, you get very negative feedback. So in the field of oculomics, let me just say this. If you start using this technology, for example, predicting and giving a risk score about a person's cardiovascular risk and asking the patient to say, hey, look, you're in the high risk group, please see your GP or cardiologist. And when they go there and they did all sorts of tests and they found that the CT scan is normal, the treadmill is normal, and the person went through a whole series of tests to say, look, you don't need any cardiovascular treatment. What is the impact of this on the Oculomics group that's doing this research, right? How does it do? What does it do to the field? I would be extremely cautious when you're looking at this. Therefore, the clinicians involved, for example, the cardiologists or the diabetologists, how would they interpret an information? I've seen in China now some technology whereby retinal photo, they give you a score and a risk of stroke. Mine is actually high, right? So what do I do? Should I go to a neurologist and say, look, I have a high risk of stroke. Can you do some tests for me? And what will the neurologist interpret this retinal photograph-based risk of stroke, which in my case is high? What would they do? Are they going to do MRI for me? Will they pick the risk that I might really be at a high risk of stroke, or will they dismiss this? I think these kind of questions have not been answered, and is a particularly challenging field in oculomics because you're dealing with another specialty and you're dealing with a specialty that is not familiar with your technology. And then finally, who's going to pay for it? So, if I have a retinal photo and I'm able to assess my risk of Alzheimer's disease and stroke and so forth, and I find that, it gives me good information, do I pay for it? Does my insurance pay for it? Does the government pay for it? How would they know it is useful and it is cost effective? I think we do not have those information, essentially. Now, the future, of course, will be things like foundational model, which I will not speak about. I think some of you are aware that there has been like the red found model, which is also useful for oculomics. And I think using that foundational model does save and does expand this research to many smaller groups that do not need a lot of labeled images and a lot of fine-tuning, because they could use the foundational model as the basis for what they would like to do in their own clinics and in their own setting. We've also looked at combined vision language models that everyone's aware of. So likely in the future, the oculomics output would not be a standard output based on the image, but it will really tack on a large language model and maybe even an interactive agent-based large language models that could interact with the patient. So the patient could say, this is the report that I have, what does it mean, right? And the large language model would have to interpret the image as well as the patient characteristics to give a personalized recommendation plan to the patient. And I think that that will be likely the future of kind of oculomics. So let me finish this talk, and hopefully that has given you a perspective of where we are at. That I think, it is the retina certainly, but other parts of the eye less likely, but retina certainly provides many signals and information. And I think that There's been a lot of advances over the last 30 years. First on the move from film to digital retinal imaging, allowing the possibility of where we are at now, which is artificial intelligence. And then, certainly in cerebrovascular neurodegenerative disease, there's been a lot of progress, but other diseases less so clear. There's also development in hardware, which will support the field of oculomics because you have smaller portable retinal cameras and even OCTs. And of course, there's software, which I think artificial intelligence, foundational models are those kind of foundational technology. And then I think it does need a lot more work for it to be used before it becomes a very interesting association study that gets into good journals, but is of little impact to a patient's health or for clinical use. Yeah, so I'd like to thank the group for inviting me to share, and I hope that you've learned something today. Thank you very much.
Thank you, Professor Wong, for this very insightful lecture, and particularly the challenges and the next steps, which really help us further enhance our study. So now we start our Q&A session. So as the moderator, I would like to take the chance to ask the first question. So maybe we are all aware that one current issue is that the ocular changes may not that specific to systemic disease, because they may also occur in normal aging and some ocular pathologies. So now with AI and deep learning, which could also be a black box, so how can we further move on and enhance the acceptance of this concept, oculomics, among different stakeholders?
Yeah, so I think let me just say this. I think we are really in the beginnings of oculomics, right? So it's good to have more people interested. NIH has a grant, and I'm sure Yali, David, and others would maybe be applying for some of these grants and so forth. But it's like genomics, right? Genomics took so long, right? If you remember the early wave of GWAS studies, every other month there was some GWAS studies in Nature Genetics, right? Now people say Nature Medicine is a hot journal. It used to be Nature Genetics was the hot journal. Everyone's submitting everything on GWAS under the sun to there. Then what happened, right? You have very little translation. There's no clinical trials. No one know what to do with it. And those association studies, which got larger and larger and larger, became of less interest. I think oculomics is suffering a little bit from there, right? We do need more studies. I'm not seeing that kind of collaborative studies. I know that Carol and others, particularly from US, are getting the collaborative groups together. It does require much better platforms. What are those platforms? The platforms will be, how do you anonymize data? How do you put it on a secure cloud? How do you use federated learning? How do you get mutual benefit? The GWAS studies was very successful because there was a steering committee, there was sharing of authorship, there was, everyone kind of agreed that, out of 10 studies, we would take lead on one, you take lead on the other, and so forth. We don't have that discussion at the moment. Kind of everyone's still hoping to be the first one to get into some of these major journals. I'm sorry that I have no camera. I would love to see all of you. I'm seeing some of it here. I know it's quite rude to not see it, but don't worry. Just it's fine. But anyway, I think I think that that's the first step. Then I think simultaneously, as I said, some people need to work on very specific oculomics. I think when we work on the deep learning for diabetic retinopathy, we were very specific and we were very clear that we wanted to push it. Many scientists, particularly biomedical engineers, do not want to move the translational pathway, right? They like to go to new areas, essentially, right? So once I've done one DR algorithm, that's the end. I'm not interested in DR anymore. But actually, what was useful is a translational pathway. And the translational pathway, many of it does require a little bit of algorithm tweaking to make it usable. I would tell you that most algorithms that I've seen, when I ask them for a usable interface, is not possible, right? They give you a code, a Docker. I mean, who's going to use it? How is it going to be used, essentially, right? So in other words, you have no validation studies. You have no validation studies. You need to be able to have something that allows people to say, oh, well, let me just give you an example. Since Lisa is there, age gap. Can I use it? If someone says, I want to use it, a single patient from Africa says, I want to know what's my age gap. How am I going to use it? If there's no commercial company, they cannot use it, essentially, right? And therefore, they don't have a feel of their age gap at all. And the commercial company, you don't have data, right? So the air dogs in China, you have no data. You know that they come out with some information. So I think the research community must be able to use downstream and must have some of their engineers and postdocs to say, I want something downstream, not just on my GPUs, right? If it's stuck in your GPUs, I can tell you no one will use it, which is a shame, essentially. So I think both sides are important: the developmental studies, the widespread acknowledgement, and then the downstream studies.
Yeah, thank you, Professor Wang. I think you set a very good example for us for the downstream applications. So yeah, Professor Jia, you have a question?
Okay, let me turn on my camera. Hi, Tien Yin. Very fantastic talk. This is my first time to see, the overview of your very high impact articles, and I see a lot of paper from Carol, from Lisa. It's really, it's a good overview for the history of this area. My question is related to OCT, actually. I know recently you really promote those integrated imaging or multimodal imaging modality. You put into the foundation model, try to use the image from the different device. But in your earlier paper, or even from other group, they mainly focus on the founder's photo. But 10 years ago, I think when you start looking at those founders' photo, OCT already widely used. They should have tons of data available. I just wondering if there is some difficulties interpretation the OCT image, or there is some difficulties on the deep learning. I just want to know your explanation on that.
Okay, I understand. So I think, Yali, you work a lot on OCT, and many groups work on OCT. I think there are two things, right? When we started, the idea of multimodal was not as popular as today. That's the first point, all right? Yeah. And therefore, we thought single modality was enough, right? Yeah, that's the first point. The second point is that the labeling of images was critical when there was no foundational model, right? And therefore, as you know, retinal photos were labeled already, right? And could be labeled, and it was easier to label it. Whereas the OCT in many of these studies, which you can get, actually Beaver Dam and Blue Mountains, they all have OCT, are unlabeled. So who's going to label them? Now, maybe now we are able to get them, and of course we hope to get them, or a lot of you would use them because you have unlabeled data, which is only possible now, right? In the last year, where you have self-supervised learning, essentially, and you can use foundation model to fine-tune it. In fact, all you need is maybe 200 labeled, right? And your model will be superb. We don't have that kind of techniques when we started. So the retinal photo was the best and the easiest, and it's still laborious, right? For us to do some of the early MESA study, it took 1.5 years to grade the photos, essentially, right? The retinal photos. So that's the thing. But I want to go back to the field of oculomics. In the development of the algorithms and technology, many people think multimodal is the way to go, which I think means that they are trying to make it complex. Now, when they make it complex, it's useful for tertiary care. So let's say I have a complicated eye problem, right? I'm going to have multimodal, right? Because I will have OCT, I might have OCTA, I'm going to look at fundus photo or clinical examination, I might need other data. But if I'm going for screening, I should make it simple, right? And therefore, it's very difficult, and I know Carol is working on that, and UK tried this, which is that I want to screen for diabetic retinopathy with both fundus photo and OCT. I mean, how are you going to do it in a primary care community clinic, right? You're adding a complexity to screening, right? So in other words, you do need to simplify for what I would say the bigger population use of screening versus what we'll say the multimodal use of artificial intelligence in diagnosing complex disease. Now, if you ask me, where is AI going to have the breakthrough? It's likely in screening, right? We know that it's going to be useful for primary care and community care, whereby it will be better than the current community and primary care, where they do not know how to diagnose many things. If you're going to say it's going to replace a specialist in a hospital, a retinal specialist with multimodal function, I think AI is going to find it a big challenge. And therefore, I think while the field is going to multimodal, the clinical use is actually going for simplicity, a single modal downstream application. So I think that this is maybe something to help Yali and others to look at this again from the technical people. They like multimodal because it's the next big thing. But from the effectiveness implementation for those working downstream, they're looking at single modality, simplified thing that provides sufficient information for screening, proving that it's effective.
Thank you, Tien.
Thank you, Professor Wong. Andreas, you have a question?
Yeah, thank you. Thank you, Professor Wong. This was a really fascinating talk. So I have a question more from the clinical side. So where do you see, or how do you see the role of ophthalmologists in the field of oculomics more on the long term? So our ophthalmologists needed to kind of exclude some confounding ocular underlying diseases, or are we needed for interpretation, or where do you see clinicians in this field?
Yeah, I don't have very much hope that they will. Let me tell you that, you know, in all these years of me giving talks before the interest of oculomics, most of the time when I give talks in ophthalmology conferences on diabetic retinopathy and AMD, right? Hardly do I give a talk on the eye as a screening for some other disease. And I've given talks to people in stroke. I've given talk to diabetologists. I've given talk to cardiovascular groups. I've actually attended American Heart Association meetings, right, in the past. They are the ones interested in oculomics because they're looking for something, because the disease areas of ophthalmologists is eye disease, right? And therefore, I'm not certain that I'm able to see ophthalmologists, besides it being of interest to ARVO researchers, it going to American Academy of Ophthalmology or, you know, URetina, for example, right? Because it is really not their business. That's the reality, right? It's really not their business. So I am thinking that the companies that wants to use it commercially would have to bring this technology to the correct groups, the diabetologists, the GPs, the so-called kidney specialists, and so forth. You know, and I think that ophthalmologist is at most a consultant, and maybe even then not that important, because we're looking at features that ophthalmologists do not need to know, right? It's not retinopathy, it's not AMD. It is signals, right? And signals could be known by the technical people that's working in the ophthalmology field, but not ophthalmologists.
Yeah, thank you, Professor.
Thank you. Thank you.
Yeah, since we are running out of time, let's have the one last question from Lisa. I saw Lisa raised her hand.
Thank you, Emma. Thanks, Professor Wong. Fantastic presentation as always. And further to Andreas' question, and I'm really interested in the implementation, and I have some experience in implementing the AI technologies in Australia context. And I find it very interesting, and also I wanted to ask your feedback on this or comments on this, because when we want to implement the AI technology for eye disease screening in the primary care settings, such as the GP clinics, as we know that 80% of the Australian people will visit GP once a year, and so this will make the GP clinics opportunistic screening for eye disease. However, when it comes to the DR screening or glaucoma screening, and they're not interested in that, and they don't know how to interpret the results to the patients. And when we implement the CVD risk score or CVD risk assessment into the AI technology, and they're very interested in that. And because it's within the business and it's within the responsibility, and they know how to communicate with the patients in terms of the CVD risk assessment and how to manage the CVD patients with CVD. But in terms of the eye disease, it's hard. And another thing is that it's an extra investment for the GP clinics, such as the fundus camera, and the technicians know of how to capture the fundus image. And this kind of extra investments into the GP clinics makes this implementation is very difficult. What's your the feedback and the comments and the insights into this scenario?
Okay, very good, very good. I think this we need a lot more discussion. But basically, as I said, I answered one question from Andreas, which is, it's not really ophthalmologists going to push it, right? Then is it the GP is going to push it? That's the second question, right? Which is what you're asking. I'm not sure that they're going to push it either. So if ophthalmologists are going to push it, GP is not going to push it, who's going to push it? Actually, the answer is basically what I think is the future. The patient's going to push it, right? And for the patients to push it, they need it like the watch that you're all wearing, counting how many steps you take, and some kind of health risk, right? And Oculomics need to go when the patients have it, then they will push that technology. Now, how is the patient going to get it is where I don't have the answer. Some people think maybe they get it in the mall or, and so forth. So I think whether or not it's an ophthalmologist clinic, unlikely. GPs, if the patients want it and it becomes commercially available, maybe the GPs will buy into it. If not, it goes to the malls and it goes to commercial shops. Now, how and when will a patient want this? It will want it when the field of oculomics are sensitive enough for them to not have single time point assessment. Now, I know that some people have already in the US taken the blood for 23 and 23andMe or something. I can't remember the company. I think some of you are aware. Which means that they want to know their genetic risk score, correct? So they're paying $150 for it because the patients are paying for it. The GP is not paying for it, either ophthalmology, insurance company, but they want to know because I might have some risk of some Alzheimer's disease in the future. No, but that's a once-off. Oculomics will happen if it is sensitive to therapy and to changes that you can make. So, for example, if I lose weight, my oculomic score improve. I would say maybe I'll have it, right? I would take it once every 6 months, right? Because how do I know whether I'm getting better? If I am on hypertensive medication, now I have hypertension, unfortunately, and I take, an ACE inhibitor. Yes, I measure my blood pressure, but it's very variable, right? Morning is a little bit better. When I'm walking, it's not so good. If Oculomics can tell me my blood pressure control over time better, I think I might use it. And therefore, it will become a tool that patients will drive. So that's the field of Oculomics, because the field of genomics is now driven by consumers, right? They are the ones that's paying for a whole genome-wide screen. One day, all of us are going to have whole genome-wide assessment at birth. That's what I say. In other words, the hospital will say, do you want a genome-wide association for your kid? We can do it in the first fingerprint. Yeah. Why do we do it? It's $150, right? That's, I think, important for the future. So Oculomics is not going to be driven by GPs or ophthalmologists or anybody until the consumers, which means the healthy people and not the sick people. The healthy people feel that the Oculomics gives them information about the health, plus the health can change when they do something about it, right? That's how we buy our watches and our sleep. It tells our sleep pattern, and we are very happy when it gives us points for that, essentially. So I think just take a look at it. I think the field is quite early, quite exciting. I think some of the younger researchers will see the fruition of this research many years later.
Thank you.
Thank you, Professor Wong.
Thank you, Professor, and thank you all for attending this. We will close this webinar. And if Professor Wong approve, maybe I will seek your approve afterwards, because I will check the video and see whether we can share it online. Thank you.
Okay. All right. I got to go.
Everyone.
Thanks very much, everyone. Okay. Take care.
Thank you.
Bye-bye.
See you all soon. Okay. Bye. Bye.
Thank you.
Bye-bye.